I did my postdoc and little bit project scientist studies with Dr. Gubler back in 2007. And then I took another position at UC Riverside to work on subtropical crop diseases. I spent quite a time there. And then when Dr. Gubler retired, this position was open and I applied, and I was lucky that I was able to get this position back. And now I'm happy and excited to work on grapevine diseases with you guys. So I am mycologist, work on the fungal diseases. When I was asked to give a talk on the fungal disease, I said yes. So that's I would like to do that. So today I'm going to give you a general information about what the fungi are very from the beginning, and then how do they live, what is their biology, what kind of epidemiology that they like, and eventually I'm going to talk about the common fungal diseases of grapevine that you can run into. First of all, I just want to start from the common plant pathology aspect. So the causal agent of the diseases could be biotic or abiotic. Okay? So among the biotic, the cause of the disease, fungus is the number one. Why? I'm going to talk about it, why the fungi are number one causing diseases on the plants. We also call them fungi. We also call ourselves that we are fungi. Bacteria is another organism. I'm sure you have heard about it. So they are the different than the fungi. When it comes to fungi are eukaryotic organisms, which means they are not plant, they are not animal, they have their own kingdom. It's called the fungal kingdom. So they have been on this earth more than 500 million years ago, before the plants were exist, which means these are the guys figured out how to survive, how to kill, how to make like the organisms to eat and then digest and then make the good for their next generation. That's all they figured out during those 2 years. So the reason that I'm telling you that they are so complex or complicated organisms that when it comes to today, it's going to be difficult to control them, because they figured out, or they will figure it out if it comes. When it comes to bacteria, they are not eukaryotic. What is the difference between eukaryotic and eukaryotic? Eukaryotic organisms means in their cell, they have a real nucleus. Bacteria don't have any nucleus. Their DNA is just spread within the cell, so they are less complicated. When it comes to virus, they don't even have like the real DNA. They have like RNA, short of the RNA. They just hang out in that organisms. The reason that I'm telling you is that controlling those organisms is gonna be more or less complex, depends on how organism is complex within this system. So there are also nematodes. I'm not going to talk about it. There are also parasitic plants that I'm not going to talk about. I just wanted to give you a brief introduction about like what these organisms are, how do they act. So this is the size comparison between the pathogenic organisms outbreaks. So this is the fungi. This is the nematode that you can see at they're very small. That means they can get into their mycelium into the cell. So that's the point that I'm trying to make here. So they are fungi and the spore-forming organisms. They can move from one location to another location. over 100,000 species exist which are saprophytic. Actually, when we call the fungi, we always like to think about the bad things. No, most of the fungi are good for the environment. What they do, they just like to eat the leftover organisms and then turn into the good organisms that other organisms could benefit. Okay? So without the fungi, the life on Earth wouldn't be possible or exist, I would say, if I say. However, 20,000 of the species are the pathogenic on crops, on plants, on agriculture, so we are going to talk about a small amount of them. So we talk about that. I want to show you that they have their own kingdom, they have their system, that I'm going to give a bit more information. I just want to show you a short video that shows that the water mold, which is group of the pathogen Phytophthora, I'm sure if you have heard about it, cause root rot. These are the group of the pathogen. They are not animal, they are not bacteria, but they somehow produce a kind form of the spores, which is called the zoospore, could swim within the accessible water under the ground, and that they can find and identify their host and then contaminate or infect. So this is the single zoospore, the sporangium, that say that they have a lot of zoospore. These zoospores, they are not animal, I said. They don't have eye. They only detect their host by when the host release some of the secondary metabolite in the soil, they are able to identify that, and then they go target it there. So as soon as there is everything is under the, these are happening under the soil. Okay. As soon as they just release from their sporangia, that's the structure, and then they detect the signals from the root, they just go there, swim there, I will say, find their host, produce the kind of like the hostorium that could penetrate into the plant. If they cannot go into the plant, they produce kind of enzyme, which is, I would say, their teeth. They can open the place and then get into the plant and then produce secondary metabolites, and then they kill their plants this shape. So they are that kind of like the complicated organisms that they could work on it. Some of the fungi are we call them Ascomycete. It's the group of the fungi. So they produce kind of like the conidia and conidiophore, these kind of structures. These are kind of their seed. Each single conidia or spores are able to reproduce another new fungus that they could cause disease. Yeast that we utilize, most of them are also part of the fungal group. When it comes to yeast, they also have different kind of reproducing system. They have the budding. So imagine like that there's a one single cell yeast. If everything all the conditions are perfect, they just start producing a small budding like that, and then divide the nucleus by mitosis, and then they just produce the new structure within a short time. So every organism that they have one goal to inherit their gene for the next generation. So that the fungi are not exception of this. So fungi are also doing the same survival mode. Today, most of the diseases of the grapevine that I'm going to talk about are the part of the Ascomycete, which is the class of the fungi. The reason that I want to show you is that these Ascomycete group of the fungi are able to produce the fruiting bodies, which we call them asexual, which is conidia, spores, conidiophore, or pycnidia. And also they are able to produce sexual fruiting bodies. Their sexual fruiting bodies produced by not the male or female, they have their mating type A and B. We call them plus and minus. So which means that two genetically different mating type, same genus, same species, get together, they share their nucleus between them, and then they reproduce this kind of structure that we call them Ascomycete fruiting bodies, and then they just spread their next generation. I want to point out one thing. Why the fungi are more complex than the other pathogens. When it comes to survival, bacteria and virus depend on their host. When their host dies, bacteria and fungi, the virus would die. Therefore, they wouldn't kill their host immediately, because their survival and next generation, everything is dependent on them. When it comes to fungi, they don't care. They are high in that complexity. They figured out how to survive. They are able to produce different kinds of the fruiting bodies, which these are the naked acai, which is one pathogen that cause Cleistetium, which is the group of the fungi caused the powdery mildew, Cleistetium. Perithecium, which is group of the Botryopsera, group of the fungi, I don't know if you have heard about it, cause. And also Apotecium, that's kind of the lichens they are produced. I'm going to talk to you about more about it. Mushrooms are also fungi. Maybe when we call the fungi the mushroom. Mushroom is the fruiting body of the fungi. Their body is the mycelium under the ground. Mushroom is tip of the iceberg of a mushroom, I would say. The most of the body on the ground, and then when the pathogen or fungus decide to reproduce, they just come and then produce a mushroom, and then that mushroom is the reproducing spores in that one. As I mentioned earlier, that before the trees were on the earth, mushrooms were around. These are like the three kind of the mushrooms. How do we know? They found the fossils of it. This is the fossils that was found in Saudi Arabia. That was the 450 million years old. And then that's the point of the mushroom right there, before the trees were around. At first, they thought that that's some kind of different organisms, but they identified what was the one giant mushroom. Let's come back to our diseases. We just too much background information. Disease concept. As a plant pathologist, when I want to have the disease on a plant, it's not easy sometimes to have disease, unless I come up with the pathogen which is able to infect the plant, host which should be susceptible to the pathogen, and I have to find the right environment. Without having any of these, you will not have disease. So everything has to come right. Because of that, when we identify our disease and then try to find out how to control it, we also look for the what kind of environmental condition that they survive. If we can play with those environmental conditions to control them, which Dr. Gubler used to come up with the Thomas Gubler model for the powdery mildew, that worked, and then that he found the right condition, right environmental for the pesticide application. So these are the couple of the slides how we identify the pathogen. I'm not going to go through that one. So what we do when we get the samples of the infested plants, we just make isolation and then do the DNA analysis on that one to identify them. I'm not going to go through. I'm just going to dive into the common fungal disease of grapevine that you are going to run into. One of the diseases is the Petri diseases. It's also caused by the Farmanella chlamydospora. It has been around for a long time. So this is the fungi that grows on the Petri dishes. Same fungus could also cause Eskalen disease, which is also another disease that causes on the mature wines. As I mentioned earlier, these are the Ascomycete. They are able to produce sexual and asexual reproduction, the spore-releasing organs. In this case, we identified their pycnidia on that grapevine wood tissues in the past. So these are the spores. So these are the spores under the scanning electron microscope, which you cannot sometimes see with naked eye. You have to use the microscope. So this is another movie shows that after the rain or any precipitation, those fruiting bodies, pycnidia, could produce or release their spores from inside to out. The reason for that, they are looking for how they can find the new healthy wine that they can reproduce and then continue doing their cycling. So this shows that how many spores one single pycnidia could produce within the structure. Eskalen is another group of the fungal diseases that you may have heard or you know about it, which is caused by another group of the fungi. Tocninia is another of the pathogen. So it is more common on table grapes because those symptoms could be apparent, but very easy to identify on table grapes than the wine grapes. So, and also it shows more in the Central Valley or Southern California than the Northern California because of the climate. Also has effect on these Eskalen group of the pathogen. They usually show up during the June when the first heat comes up in California climate, you know. And after that, you will see the tip dieback or tendril dieback, as you can see here. Those are the first symptom of the Eskalen disease that you will run into. Later on table grapes, you can also see this kind of tiger stripe, kind of the symptom, which is very common symptoms. These are the symptoms on the fruits. So it used to be known as a measles. They look like the measles. In studies that we have done in the past, when I was postdoc in Dr. Gubler's lab, that we were thinking whether the pathogen is coming from out, entering into the berry, and then causing these specks, or coming somewhere else. We were able to identify that, in fact, some of these fungi are vascular fungi, which means they live within the wood tissue, xylem tissue. While they are living there, under the conditions, right conditions, environmental conditions, they reproduce secondary metabolite, which is kind of toxin that could cause this kind of symptoms. Same pathogen is able to enter into the plant or into the fruit through the stroma or lenticel, which are the natural opening for the breathing for the plant. They can also get into plant. So those are the two things that I just want to mention. So nobody would like to buy this kind of fruit when you have it. I mentioned that the vascular pathogens. There are a lot of pathogens that they attack or affect a different part of the plant. These are the vascular that they like to live within the woody tissues. So these are the common symptoms. So you see that this is a close-up picture of these vascular tissues. They're like the black, they're coming. It used to be known, this disease used to be known the black goo, because of that small structure. That small structure is response from the plant. You know how we have the allergy, when we have the reaction for the allergy? Sometimes some people have too much reaction to allergy that could hurt them. Plant has the similar reaction when they get infected outside organisms. In this case, when the pathogen gets into the xylem tissue, plant recognizes to prevent the movement of the pathogen, they produce tyloses and gumming, that kind of like the gooey structure to prevent their movement. But they don't realize that that plugging also prevent them to get the water and nutrition from the roots. They kind of overreact, and then that causes the dieback on the plants. So if we could tell them like, hey, do a little bit yoga, relax, it's gonna be okay, maybe they will be okay. But that's like the things that they react that kind of disease. There are Basidiomycete group of the fungi, which is mushroom, same class of the mushroom. They also contaminate or infect the plant, then they cause the rot, soft tissue. Again, they cause the soft tissue. They also contribute to the ASCA group of the disease complex. As you can see, when I say the ASCA and young vine decline, I'm not talking about single organisms. There are many organisms that are contributing to the ASCA disease complex. So it's the complex of the disease. That's why we were not able to find the silver bullet to control the disease path, because it's a very complex disease. More than one pathogen, different organism could cause it. These are the different morphology in plant pathologist perspective. They look beautiful in the plate. Again, in the past, I talk about the sexual reproduction organs of the fungi, Ascomycete. These are the parasites. It's a very small, the flask shape, the structures. They are filled with the spores, which we call them ascospores. I just want to point out here is that the structure is organized very well in old pruning wounds, that their neck is facing to where the light comes from. What does it mean for the structure? For the structure, wherever the light comes from, they can release their spores in the air. That's how much they figured out. So when we have the rainy condition, because their neck is not facing the down in the grapevine, they want to go in the air because they know from the air they can go and then find a new host. That's what they do. I just want to point out that every single neck of these that you can see, they all are facing the light. That's very good. So what they do? What does it mean for them? After any precipitation, keep in your mind, I don't say rain, I say precipitation, which means any kind of moisture, high humidity, or fog could also initiate or activate these fruiting bodies, not only rain. So, Eutypa Dieback. When it comes to Eutypa Dieback, they are pathogen, but they are different than the ASCA group of the vascular pathogen. They are not vascular pathogen, vascular. They are canker pathogen. They cause canker. What is that? That means wherever they colonize, they kill the tissues. Which means when you have the canker on your plant, there is no way that you can recover back. That tissue is dead. So we are going to talk about it, how to control canker pathogens because of that. Colleagues of mine, Dr. Fleur Troyes, who has worked on this Eutypa the dieback in the past, he also identified their fruiting bodies on the trunk of the plants. So they produce the asci and ascospores. He also identified same pathogen on different hosts. Guess what? Most of these pathogens, they are not only depend on their one host, they also specialized to colonize on other hosts because that's the good survival method. What does it mean? Which means it doesn't matter sometimes when you clean up, keep everything clean in your vineyard, the source of the inoculum may come from the other native trees around your vineyard. That's could be the possibility. Autospheric canker. Bud canker is another group of the pathogen. They also cause canker. So this is also used to be known as dead arm disease. When the pathogen gets into the plant, mostly from the pruning wounds, and then they just move back and then cause a lot of diseases. Before we go further, I just want to show you a couple of the spurs that I cut it longitudinally to show you how the canker pathogens were able to get into the plant from the pruning wounds. So I'm just going to give you from here so you can just pass from that. Don't take it home, okay? I need them. I need those diseases. So you can see look at that old pruning wounds, how that canker pathogen was able to get in down on that area and everything. Speaking of fruiting bodies, the fungi that produce, I'm also going to give you a couple of samples that this is the one old pruning wounds that we identified, Neophysococcum parvum, which is a very aggressive pathogen on grapevine. This is another Autospheria group of the pathogen, Autospheria dotidea, that was able to get into the stem of willow, which is very common riparian trees, and then they cause the canker. Then they form pycnidia and parathecia on it, and then they are ready to go after any rain event. Canker, when you look at the inside of it, it looks like the canker that's caused by the Eutypa lata, which is very common, that it's difficult to distinguish between those canker pathogens. Here's another short movie that I would like to show you, how these parathecia or flask-shaped structures could release their spores. Different than pycnidia. As remember that pycnidia was just oozing out and then staying around that pycnidia, right? Let's take a look at it. What's happening when it comes to parathecia? This is the group of the parathecia. It's the cartoon. This is the structure. They are very close structure. There are a lot of asci. As soon as they get that moisture, they just start expanding and then build a physical pressure within the structure. That pressure cannot stand longer, and then they release their spores. Right now we are in the structure. They just release their spores in there. What does it mean? They are so small. As soon as they are in the air of the vineyard, they are going to be picked up by the wind, and then they can go further distances. If this could happen during the pruning season, all your pruning points are going to be subject to the infestation of these things. So that's why it's important to know biology of these fungi, epidemiology of them, when do they, under what condition they release their spores, so we can get an idea how we can manage them without applying all the pesticide, or how we can manage them by cultural practices to reduce the pesticide application. So, because at the end, as a farmer grower, you want to grow your grape without any diseases. I mean, who cares if you want to do the cultivation or other things? The goal is at the end have the good amount of the yield, right? So, but if you want to do sustainable bay, which means if you want to get the same yield every year, so you gotta think about the cultural practices and combine with the chemical applications. It's called the integrated pest management or integrated disease management strategies that I'm going to talk about. So in the past, we also did some the spore trapping studies during the year. We identified that these two graphs are simply showing that these spores are releasing during the precipitation, which these columns are the number of the spores that we trap from during the years each month. So the red lines is representing the temperature. The blue lines are the precipitation. So these bars, number of the spores. As you can see, during the winter, there are a lot of spores in the vineyard, in the air of vineyard. There is no precipitation, no fungi, and then when the rains comes. I just also want to point out that there are a few fungal spores were trapped during the summer without any precipitation. That could be because of some of the leftover, the old branches could contain some of those fruiting bodies. They could be activated by the irrigation. So that's where the vineyard sanitation is a very key role in here. Well, since we are talking about the fungal diseases, we cannot pass the powdery mildew. Powdery mildew is another very important fungal disease of the grapevine. So these are the symptoms if you haven't seen it. So there are a lot of ways of controlling it. So nowadays we have been seeing some resistance against some of the strobilurin group of the fungi, and therefore it's very important to combine the pesticide or fungicide application with the other practices as well. What I mean is that if you can find combine with the other application to reduce the pesticide application, that would be the goal. So there are a lot of information out there. So Dr. Gupler have been running fungicide screening trial, which I'm going to be taking over that trial and then continue doing it starting from this year to following years. And then all those information can be found online either in my lab website or go to Department of Plant Pathology website. If you don't know, I can give you that information too. I just want to mention about the botrytis bunch rot, which is another the pathogen that has a wide host range. It causes the diseases, especially during the rainy and wet season, like this year would be, and they cause the diseases. So these are also group of the Ascomycete. They produce a lot of spores that they can release during the precipitation. They are not only causing diseases on grapevine, they also have other hosts, which means they can also survive on other hosts for a long time. So these are a couple of examples for the botrytis scenario. It's very interesting that we call the fruiting bodies of these fungi are as overwintering structure, which means when the winter comes, they produce those structures that they can survive, and then spring they can come back and everything. So botrytis is the kind of the fungus they kind of like the has more tolerant to the colder temperature than the other fungi. So they could also cause problem in cold storage too. So it's very important if you are the storing your fruits in the cold storage has to be cleaned up, disinfect once in a while and everything. So these are the conidio. They are the conidiophore. This is like the structures. They produce like the conidiophore. It's a crazy way that a lot of conidiophore. So this is one of the publication that I found in 2016 that shows Botrytis cinerea, which is one of the one that caused the rot on grapevine, is able to grow in all 0 Celsius, this is the Celsius degrees, to the 25 to 30 Celsius. My point is that they can also survive in the cooler temperature. My colleague, Dr. Jose Ramon, also identified that the Promopsis dieback could also cause canker disease. We used to know the Promopsis only cause this kind of like a shoot dieback, as you can see. He identified that also could cause the canker further and then dieback on the plant. So Promopsis is another fungal pathogen. It's very common in California, especially during the wet years. So these are the symptom on the leaves in early stage. These are the symptom on the leaves in the late stage that you can see. It can cause the speck on that one. How am I doing with my time? 5 more minutes. Okay. So these are the pictures from the Jose's the publication. So he did a very good pathogenicity test to show these Promopsis could also cause dieback on plants. Armillaria is another pathogen that is also known as root, like the oak root fungus. It's also known as a secondary. When you have the root are damaged with something, they can find easy way to get into the plants. Okay. I'm just going to talk about the management strategies of these fungal pathogens when you are establishing a new the vineyard. You gotta get the clean plant material. So this is the one of the nursery bed. The plants don't look good in that nursery bed already. So when you look at this kind of the roots, they don't look good either. Sometimes I have heard over the grapevine that when some growers could get the good deal from the nurseries, the cheaper price, because when they have the not good growing crops, they could sell. So no cheap deal is the good deal. So you gotta be make sure that you are getting a healthy, good rooted plant material. If you are a nursery, I'm just gonna give you some of the recommendation that to apply some of that aspect to have the healthy, good growing nursery plants. This is the couple of the samples from the nursery plants that they also show another discoloration. If the callusing hasn't been done properly at the bottom of the cuttings, what we are doing is we are just planting another cut wound on the ground and then abandoning them for the soil-borne pathogens that could get in. So that's very important to have a good callusing at the bottom of each nursery plants. Those are the things that I would recommend. Some of those Pycnidia fruiting bodies could also show up on those plants as well. I'm not saying that what we have been doing in the nurseries in California, these are the key point that you gotta be thing. So these are the not good plants, which means that these plants stay in the pot for a longer time. The plants start producing the J rooting on the plants. Okay, so these are again not good to have that kind of bottom of the not good callusing. This is better, not good callusing, and then you can see that kind of discoloration in that one. Okay. Management in nurseries. How do we manage? In nurseries, hot water treatment is the one of the aspects. But there are some studies that show that the hot water treatment in 122 or 50 Celsius degrees is not enough to control some of those Eutypa and Autospheric group of the pathogen. They increase it. It is an option, but it is not 100% clean. You have to rotate the nursery beds, because when you have the pathogen or something not good for the plant, they can accumulate within the nursery beds. Those are things. You have to treat the pruning wounds of the mother plants, because that's what they are also suspect. They can also be infected by the airborne pathogens as well. They could be susceptible. So you have to apply the good cultural practices, not too much water, not too less water, to stress those plants to environmental condition. And then you have to check the nursery beds periodically to identify what it is. Management in the vineyards. Okay? You have to start from the clean, the clean plant material. Apply good cultural practices to minimize the stress, because stress indicate that some of those latent pathogens, which pathogens could enter into the plant and then wait until the right condition comes, and then they can just colonize on the plant. So you got to wait that long time. We now know that after the precipitation, there are a lot of spores in the air of the vineyard, so you got to protect those pruning wounds. Delay pruning is recommended if you could. But I have been seeing and hearing and everything, so I think it's the labor issue. So when you find the right labor in the right time, people just go for it. So they cannot wait until the late pruning. If applicable, consider the double pruning. Depends on your trail system or the conditions. So double pruning has shown by Dr. Weber that also could work on these grapevine trunk diseases. So double pruning is simply what you do. It could be mechanical. If it is applicable, you go and then remove the excessive branches about like the 1 foot above, leave it there, and then come back in the late season and then do the second pruning to reduce the labor intensity and then cause. So in the study that was done by Dr. Weber, they artificially inoculated these spores with the pathogen Eutypa, and it was not able to move more than a few inches below. So it's recommended that's working. But it's labor-intense study. Mechanical pruning also could cause this kind of like the not clean cut surface. If the surface of the pruning wounds is not clean, it's going to take time for plant to recover, heal back with the callusing, which means that pruning wound could be susceptible longer than pruning wounds that had like a clean cut. So I believe you have been applying a lot of like the mechanical pruning, so make sure that the equipment is blade and then working properly to get as much clean cut as you could. That's like this. Spraying those the pruning wounds with the fungicide. Dr. Gubler has done a lot of fungicidal work. So Topsin M and Raleigh combination it was shown that when you apply them with the tractor, with spray, which both of them are registered in California to spray, so that could be another option for you guys. So I think I'm done with my minutes, but I just want to show you that. I'm sure, I don't know if you are able to access to this UCIPM website. It's a very good source of the information. Those are who is not a part of the UC system. So when you go to that website, you can go to find the grape, which if you are interested in, there are other crops as well. You can get all kinds of information, pests and diseases, if you don't know what it is. It's a very good, useful information. So, okay. Do I have time for questions?
Yes.
Yes, I do. All right.
The question I have is, during the time in terms of for grading cuttings at a nursery level, so if you were to grade cuttings, is it recommended for the prior to hot or after hot water treatment to actually protect the ends of those cuttings as well? Like dip them in some fungicide to kind of protect the ends so that in the future when they go into cold storage, coming out of cold storage, nothing enters in?
That's a very good point. I just proposed a proposal to do more work on that one. So what we would like to do is that when you have the hot water treatment, you get rid of most of the organisms, including some of the beneficial organisms that I didn't talk about it. There are in the plants, there are some beneficial endophytic organisms, like how we have the gut bacteria that help us improving our digestion, or also improving our like the tolerance to the diseases. Plant has the same thing. With the hot water, negative effect of the hot water, you are also getting rid of those beneficial organisms. So I'm just going to work on reloading, finding a way to reloading those endophytic organisms that we are working right now. Second one is that yes, some of the fungicide work has done in the past. There are some the fungicide that you could apply. So I would like to do more work on that one as well. So I cannot tell you the A, B, C, D fungicide right now, but we are going to work on that one.
What's the recommended, storage for cuttings? Then if I were to leave them there for a year, sometimes we'll order cuttings from nurseries. They'll be in there for 1 year, 2 years, or a year and a half, and then we'll go up. They'll send them to us to go graft in the field.
Sure.
And then, we recommend to do the firewall treatment again prior to taking it back giving it to us. Am I doing it? Am I be recommending get the hot rod treatment again prior to sending it?
I agree. You are right. Yes. So I'm not aware of any registered fungicide that could work on. That's why I would like to do more work on that one. I just proposed, and I'm going to be doing some work on that one. Very good point.
We're running a little over time. Are you going to be able to stay through the break?
I will.
Okay. He'll be able to stay through the break.
Come find me.
I know we all miss Dr. Gubler, but we're in great hands with Dr. Eskalen. Let's give him a round of applause.